The overall goal of this procedure is to electroporate charged macromolecules into craniofacial mesenchyme. This is accomplished by first positioning an anesthetized stage 28 embryo within the electroporation chamber. The next step is to microinject the solution containing charged macromolecules into the craniofacial mesenchyme. This is quickly followed by application of an electric current, thereby introducing the charged nucleotides into cells. The final step of the procedure is to visualize the electroporated tissue by fluorescence microscopy. Ultimately, results can be obtained showing changes in craniofacial tissues. These results can be analyzed in vivo or in fixed tissue using fluorescence microscopy or immunohistochemistry.
The main advantage of electroporation over existing methods like microinjection and mutation are tissue and temporal control of our molecular tools. Demonstrating our procedure today will be Jackie Tabler, a postdoc in my lab.
The pair of L-shape electrodes required for this procedure is made from high-purity 0.4mm tungsten wire. For each electrode, first cut 8 cm of the wire. Then, using putty, such as blue tack, affix a 1ml syringe at the midpoint of the tungsten wire, leaving 4 cm of wire exposed from the tip of the syringe. Bend the tip into an L shape 1 cm from the end. Trim the tip so that the end measures 0.5 cm in length.